Hereby I open this academic ceremony in which Alessandra Pizzuti will defend the academic thesis distinct and complementary insights on bistable perception in humans using ultra-high field MRI across multiple spatial scales. Before starting with the presentation, I would like to first welcome the audience here present in the aula and also online. Un benvenuto speciale pure alla famiglia. Okay, so may I invite you to present a summary of your study and the conclusions of your thesis.
So, good morning everyone. Dear Prorector, member of the Defense Committee, supervisor, family, friends, people watching online, thank you very much for being here today. It's my pleasure to discuss the content of my PhD thesis in this academic session. I would like to start with a very short experiment. Please look at the center of the screen and focus your attention on the red dots over there for a few seconds. Do you see the square moving? If yes, where are the squares moving? Maybe some of you might say that the squares are moving right to left and left to right along the horizontal axis, while some other of you can say that the squares are actually moving up to down along the vertical axis. There might even be a third subgroup of you saying that the squares are actually changing their direction of motion while I'm speaking, switching from the horizontal axis to the vertical one, and so on and so forth. What I have been showing you here is an ambiguous stimulus. It's a visual illusion. The squares are not physically moving on the screen, but they are just appearing in a periodic way that induce in your brain a perception of motion. So, if you would like to project on the screen what your brain is perceiving while watching to the ambiguous stimulus, it will look like something like this. So your brain perceives motion, and the perception of motion is changing over time, alternating between the two interpretations of it. So this illusion is also called the bistable motion quartet, as it induces in your brain a bistable perception. And this is very exciting for us as a neuroscientist, because this bistable stimulus reflects how the brain makes sense of the external world. Therefore, understanding this phenomena will get and bring us closer to understand how the human brain works. But where is this perception encoded in the brain, in the human brain? Well, the brain is a very complex organ that has multiple levels of organization. And one way to go about it is actually going across multiple spatial scales. And this is the framework of my PhD thesis. The coarser scale we define here is called the macroscale, where the brain feature appears spanning over several millimeters and centimeters. Here I'm rendering for you a gray matter surface extracted from a human brain. On this cortical surface, we can define the cortical areas, indicated here in different colors. Each area of the brain has a specific anatomical structure and is devoted to a specific function, like for instance, the visual areas that process our external input are here located in the back of the brain. But when the brain is acting, all these areas do not work in isolation, but they cooperate in such a way that they create a cortical network. For instance, while I'm speaking, there is an attentional network, hopefully, that is helping me to be focused during this presentation. Next, we define here the mesoscopic scale that allows us to zoom into one cortical brain area, look at the gray matter and some detail over there called cortical layers and cortical columns. Those features, to be revealed by us, we need at least a resolution, spatial resolution, that goes below 1 millimeter. That's the thickness of your credit card, to give you an idea. So the cortical layers on one side, in a very simplistic way, they handle the local processing of a brain area. There are layers that are devoted to receive the input, some other layer to do some computation, and finally layers that send out fine and away from this area to the next one. On the other side, we can also define cortical columns that are pillars of neurons that are highly specialized for a feature. For instance, if we talk about motion-sensitive areas, their constituents are cortical columns that are tuned for a specific direction of motion, as shown over here. Finally, we can zoom one step further and try to reach what we define here, the microscopic scale, where we need at least a resolution that is below 1/3 of a millimeter, that allows us to look even further to this constituent of the mesoscopic scale, that are, for instance, neuronal population, excitatory, inhibitory, and all the microvasculature that supply energy to such neurons. So with this, I would like to say that going across multiple spatial scale will allow us to gain insight both into the structure and the functioning of the human brain. So throughout my PhD, I have been using massively the magnetic resonance imaging, or MRI, to look into the human brain. But specifically, I've been using ultra-high field at 7 Tesla MRI that help us with its increased field strength, signal-to-noise ratio, to span a much wider range of this spatial scale. The MRI allows us to look into the human brain in a way that is totally non-invasive, and we can look both at the structure but also to the function that is indicated here in warm colors. When a brain area is active, the oxygenation that is within the vessels surrounding those areas, it changes, and it gives rise to a contrast that is called blood oxygen level depend, or BOLD fMRI, that allows us to pick up where the brain is active while we are doing a task in the scanner. So, in this thesis, we will take a small journey across multiple spatial scales. In the first part, we will focus on the bistable perception and try to look for neural correlate both at macroscopic scale and the mesoscopic scale. In the second part of the thesis, we will focus on some fundamental building blocks that are defined both functionally and anatomically for a selected brain region that are crucial for the bistable perception. So let's get started with the first part. So in chapter 2 and chapter 3 that I'm presenting you here, we did an experiment where we collect images from 10 healthy adults at 7 Tesla fMRI. This was a deep sampling, meaning that for each of these adults, we collect between 6 to 8 hours of scanning of corresponding multiple days and sessions that allows us to have a data set that is comprehensive of the whole brain, but also to zoom in, have a partial coverage, reach the sub-millimeter scale, and understand what is happening at the mesoscopic scale. And we basically study their brain reaction to the bistable motion quartet. So at macroscopic scale, to grasp what is happening at the level of the whole brain, we did use a physically inspired computational model that is called the arrow of time. This model successfully could grasp how the brain needs to reorganize in terms of its hierarchical structure in order to support a very complex task that is, in this case, is the bistable perception. But we did one step further, and we tried to understand where this complexity comes from. Therefore, we split the perception phase while you are watching to the bistable motion quartet in two phases. There is a moment that your brain starts perceiving a motion type that we call here perceptual transition, and there is a moment when your brain is keep being focused and perceiving the same motion type that's called sustained perception. And by using some connectivity analysis, we could reveal that there are two different connectivity patterns supporting the two phases of the perception. Therefore, from this study, we did learn that the entire brain needs to reorganize himself dynamically to support this complex task and change its connectivity on the fly to make this perception happening. In the counterpart, the mesoscopic side of this study, we did zoom in into two specific brain areas. The first one is human motion-sensitive areas, is a complex of areas that respond to motion. And that's called the HMT+. And as I indicated before, this area has its own constituent that are cortical columns, that they are finely tuned to different directions of motion. And as in the case of the bistable perception, we do perceive horizontal and vertical. Here we talk about two motion clusters, the horizontal and the vertical one. And this area has been already investigated in the past and found to be very crucial during the bistable perception. But together with this now, we even look into the primary visual area, V1, that is the first cortical station that processes the external visual information, that even though it has a different functional organization, we can still define at mesoscopic scale two clusters. One that relates to when we are perceiving the horizontal motion, and the other one that relates to when we perceive the vertical motion. And we try to understand their organization and communication at mesoscopic scale. So the first question we ask here is, what is changing in our brain when we are having a perception of motion versus we are seeing a real motion on the screen? And to answer this, we look into the laminar profile of these two clusters of interest of these two brain areas. And we did find that for HMT+, nothing is changing. Here on the x-axis, we have the cortical layer, and on the y-axis, the person signal change. But the two conditions appear to be the same. Differently from what is happening in V1, well, the two cortical profiles are very different between the two conditions. So it means that V1 has to go through a laminar reorganization to support the perception instead of physical motion. Then we did ask another question. While we are having a perception of a motion type, let's make the case of a horizontal perception. Is the cluster linked to the unseen motion active? And to answer this question, we look into event-related average. Basically, we follow the time dynamics of these clusters separately over time. And for HMT, we did find that our results support the computational model that has been already described previously in the literature, in animal, that there is first a neural competition between the two clusters, and then the cluster that is enhancing our perception of motion wins over the other one over time. Differently from what is happening in V1, both clusters goes up and stay up all the time. And this result let us speculate a little bit on the nature of these two activation sites. Maybe there is a double feedback occurring to V1, an excitatory one that enhances the activity of the cluster that is linked to the perceived motion, and an inhibitory one that suppresses the activity of the cluster linked to the unseen motion. So in this study, we did learn that the mesoscopic organization of these two areas of interest needs to change to support the bistable perception versus a real motion. In the second part, we start first with the functional building blocks in HMT+. So again, we looked into this motion-sensitive area. And as before, I indicate that there were two columns, one horizontal and one vertical. However, HMT+ has many other columns, each of them that respond to a different direction of motion. And this property has been already found and discovered in 80s, both in monkeys and in cats. But how can we acquire and measure cortical columns in humans? To answer this question that has been already addressed in the past, we still did collect our fMRI data from 5 healthy adults using a paradigm that could map 4 different axes of motion with these moving dots coherently along the 4 directions. We collect data at 7 Tesla, but this time we did use a different contrast mechanism that's called vascular space occupancy, or VASO, at some millimeter resolution, that compared to BOLD is supposed to be much more specific to the microvasculature and therefore to the neuronal sources. And together with this, we also did develop a series of new analysis tools that help us to quantify how columnar is the response in this area of interest, and how sensitive and specific are the two contrast mechanisms to study such a function. Well, on average, our results shows that there are axis of motion columns in HMT+, but they do span only the 40% of the depth, and not the entire depth, as it was expected. And we did find that and confirm with quantitative measure that VASO fMRI results are more specific than the BOLD fMRI. Therefore, in this chapter, we did provide a new framework that is comprehensive by both the acquisition and the analysis methods that are new and essential to us to address a question whether the brain is columnar or not in general. In the final chapter of my thesis, we did move to bridge and reach what we define as a microscopic scale. In order to do that, we did use a publicly available dataset that is composed by a post-mortem human brain that has been imaged both at 7 Tesla, but also under a microscope. We combine it with data from a living person, with one adult, again at 7 Tesla, and we did develop a series of analysis tools that can handle the multimodality of our data. But for the sake of this presentation, I will only focus on the microscopy. And this dataset comprehends very high-resolution images at 20 micron that stain for different features of the brain. For instance, one population of neurons called the inhibitory pancrevalbumin neurons, fiber density and neuronal density. And we look how this property change over cortical depth in multiple brain areas, visual area. And we did find that the laminar profile of parvalbumin is actually the most discriminatory feature across the 4 visual area. Therefore, we conclude that this information can be essential and to create generative layer fMRI models that are more accurate to detect and predict the neuronal source. And with this, I would like to conclude and say that through all this thesis, I hope I provide you some distinct and complementary insight that help us to understand the human brain perception. And with this, I would like to thank you for the attention and give the word back to the prorector.
Thanks for this beautiful presentation and overview of your work. So the opposition will be opened by Professor Dr. Federico De Martino, Professor of Sensory Circuits and Neuroimaging at our Cognitive Neuroscience Department at Maastricht University.
Dear candidate, first of all, I would like to congratulate you with this thesis. It's a very comprehensive work, and it's rare to see that a thesis is so broad, but at the same time focused on a topic and stays to the topic and actually goes in depth in it. It was a pleasure reading it both during the assessment part and also now that I had to come up with some questions. My congratulations, of course, extend to your supervision team that obviously has had an impact on all of this, and to your family that has come from far to witness this wonderful day. Now, of course, I had to prepare some questions. They may be due also, the confusion of them may be due to the fact that I prepared them while flying from the other side of the world, so maybe a bit of a complicated thing, but let's see. In your introduction, at the very beginning of your introduction, you state that when looking at a beautiful piece of art, we can recognize its intricate shapes, colors, and techniques, yet we may not fully understand why it has such a powerful impact. I argue that 2 scientific approaches, reductionism and multiscale analysis, can guide us towards an answer. And I think it's not easy to disagree with this type of reasoning, but I will try to challenge you a little bit. Let's start with the reductionist side. So you used bistable perception to understand feed-forward and feedback processing in visual cortex. I'm an auditory person, and there is bistable perception in audition as well. The closest that I can think of to your paradigm is the galloping illusion, where 2 tones are presented alternating from each other, and some people perceive them as a galloping and others as 2 separate streams. And this percept, in fact, switches constantly, and in that sense is very similar to what you have presented. So in the concept of reductionism, I would think that then whatever insight you have gained now should generalize to what the mechanisms are for auditory bistable perception. So can you tell me, if I were to now start a study on auditory bistable perception, what should I bet on? What is the mechanism that you think explains this percept at the various scales?
So, highly esteemed opponent, thank you for the compliments and for the question. This is a beautiful question. So, I will say that indeed, talking about bistable can involve different senses in the brain. And in our case, we did focus on vision. And in your case, you want to focus on the auditory cortex. So one first thing that I will do is to check which are the component or the constituent of the auditory cortex. So I know that there is a tonotopy, so we can define different tones. If these tones are actually useful for the paradigm that you want to investigate, then I will start with that and try to set up a basis. Okay, like this area is constituted by this element. Then I will still go through multiple scales, and I will do an experiment that involves the whole brain to try to understand globally what is happening in the brain. If there are other brain regions involved, and I will guess yes, so we need to find them. And then we need to zoom into the brain, and we can reveal the next level, that is what is happening at the mesoscopic scale. And there, you can, of course, look into cortical layers and the local circuitry and see how it changes versus having a real tone that doesn't provide you the illusion, so the closest control that you can have. And yeah, so already here, you might be able to see how this phenomena is encoded in the brain at two different scales. So basically, I'm telling you that I will follow what I did in my thesis because I found it very useful to look at these two different scales and then try to bridge them together. In addition, you might even want to use some computational model that can embed the result from the two scales and see if things fit together. Or, as an alternative, you might even want to maybe have a theory that like a brain theory, can be predictive coding, can be some other theory of the brain, that can tie together the result that you can get at different scales.
Okay, so let's say I do this experiment, but if we are thinking about reductionism, and if this approach of studying one mechanism in one way, it's sufficient for us to understand, do I actually really need to do all of this? Or is the mechanisms that you have, in fact, highlighted in your thesis, you think it generalizes? So why bother doing the auditory studies?
Yes, I mean, I will still do it because, as maybe I even introduced in this presentation, each brain area has a different mechanism to work, both from the anatomical side and the functional side. So what I did discover in the visual system, and specifically when we look into very detail in V1 or HMT+, and then in the microscopy part, I will just keep it on the side for now, they have a specific organization that we talk about, like columnar organization, retinotopy. So I think what I meant by using a reductionist approach is more general. It's a different level. So we can decide to look at the art piece, right? And meaning that we can look into the human brain, but the human brain is very complex. So somehow we need to cut it down to do some research that gave us some insight out of it. And I think reductionism to me means that we just focus on one process of the brain. But doesn't mean that that process will be the same for different like sensory areas.
Okay, thank you very much. If there is time, I will ask one more question. Otherwise, I'll very, very short. So if it has to be very, very short, very, very short answer to, in chapter 5, you looked at parvalbumin interneurons.
Yes.
Can you just tell me why parvalbumin and what do you think parvalbumin does?
Yes, so parvalbumin is a very special type of neurons that likely they were collected in this data set that I did use. So I had parvalbumin. They had an inhibitory neurons. So the function of an inhibitory neurons is to regulate the excitation of the other neurons. So the fact that parvalbumin, so one type of neurons, is changing across cortical depth and is changing across different brain areas should ring a bell and say that maybe when we do our computational model, generative layer fMRI model, for instance, the model from Kamil Uludag and Martin Gaulicek in the past, they were assuming that there is only one coupling, that it's the same across multiple layers. There is one excitatory neurons population and the inhibitory one. But with this result, we are proving that this parvalbumin are changing. Parvalbumin constitutes the 40% of all the inhibitory neurons, so it's the majority. And they are the neurons that are, together with another type as well, associative vasodilation, but they regulate the flow. So they produce a vasoactive, they have a vasoactive function that let being them very, very crucial for the fMRI, because maybe we can tackle them. So already the information we provide in chapter 5, how those neuronal types changes across cortical depth, I think would be very useful to embed into such a model to predict then the neuronal sources of a layer fMRI. Maybe not that short.
Thank you very much.
Thank you.
Thank you. The opposition will be continued by Professor Lars Mukli, Professor of Visual Cognitive Neuroscience at the University of Glasgow.
Yes, dear candidate, thank you very much for the nice thesis and discussions that we had last year on the conference. I really enjoyed reading all the different chapters. I really embrace very much the idea of multiscale neuroscience, and in particular the chapters 2 and 3 are very dear to my heart because I've worked with these paradigms as well. And so I will ask you questions about this, but I also want to mention that discussions, your first chapter and your introduction, I thought was quite interesting, and I bought my own copy of the Gödel-Escher-Bach, which came also from discussions with Rainer. So I'm working through this now. But my question, if you summarize, so you looked at switch and blocked responses in chapter 2 and 3. So when motion is represented and when the perception switches from the one to the other interpretation. So my question is, how do you now think does these switches come about? How is motion presented? And how do the switches start? Is it consistent with these early ideas from Leopold and Logothetis, I think, and others, that there are representing neurons that then start to fatigue and are in competition with one another, so more a bottom-up approach? Or are there other mechanisms that work top-down to decide which percept dominates?
So, highly esteemed opponent, thank you for the compliments and for the question. So you have been asking me which is the mechanism that I believe being behind the reason of a bistable perception in humans, so in the experiment. And you also mentioned that there are two phases of the perception. So let me first start with what I think about, in general, this bistable perception. So in chapter 3 of my thesis, when we zoom into the mesoscopic scale and we look how HMT plus change and not change, I would say, between the real motion and the not real motion at the level of a laminar profile, but also when we look into the event-related average. So what we found there is that both clusters, they goes up together, and then the cluster that support your perception stay more active than the other cluster. So I do think that actually these results support the neural competition models that was established at the neuronal level and predicted from the animal literature, as you mentioned, from Leopold, Nico Logothetis in the past, and so on. So I do think that HMT has a crucial role in detecting and deciding what we are seeing. So I think that indeed, due to fatigue, at the second cluster, so let's say we start our perception, there is a first cluster that goes up. And this happened just by chance, because neural competition is supposed to start as a random process at the beginning. But then over time, this cluster gets fatigued, indeed. And then it's like switched down, like it goes down. And the other cluster like wins over. And this mechanism is repeated over and over. So I do think that the bistable, maybe it's a bit like too speculative and a bit strong, but I do think that these come from actually HMT plus, for also in a series of from the clusters, like the motion clusters in HMT. I think that they code actually for what we are perceiving. Even like these results have been supported from chapter 2, when we look into the macroscopic scale. And for instance, during the switch moment, we do see that there is an interaction with the frontal area. But by computing some connectivity analysis that can give us information about directionality, we could observe that from our result, of course, they are empirical, not fully generalizing, but we did see that HMT plus starts earlier. So it's driving the frontal area. And these results actually support what we are just discussing. On the other side, when we look into the perception, so the sustained perception in the long blocks, we do see an interaction that is unique with the intraparietal cortex. So I do think that, and in that case, we found that intraparietal is kind of driving like the also other subcluster in HMT+. But I think that's come from the attentional network. So there is something that indeed the choice has been made, right? But during the perception, the sustained one, we still need some feedback that help us to keep the perception on. Yes.
Very, very comprehensive answer. I was getting to the frontal interaction, and that's you have already answered this. Very, very nice. Thank you. I have a little bit more question about the layer result. Do you, the idea exists to say that in the superficial layer, prediction errors are processed, and in the deep layers, the internal representation. So do you think your data speaks to those models?
Yeah, so this is a highly debated, of course, point of the thesis, this laminar result that we got. And first of all, I would like to acknowledge that we did use gradient echo, EPI bold. So we are limited by definition by the draining vein. So my laminar profile, actually, they look increasing from deep to superficial layers. So, and we did expect to actually have a similar mechanism between the feedback and the feed-forward condition. And by that, I mean that during the feed-forward is when we are seeing the motion, versus the feedback is when we are perceiving the motion. So when we build the experiment and we compute our laminar profile, we were thinking that, okay, we can do the subtraction. That is the simplest things, just to see our result and see if such a property comes out from the data. These deep layers or superficial layers is still debated, right? However, our empirical data show that by its art, the draining vein is changing between the two conditions. It was surprising for us to see that during the feedback, there is actually a redistribution of activity. So I couldn't do this subtraction. I couldn't use any biological model to do correction, like Aulicek and stuff, because at the end the parameters were set for the feed-forward condition and not for the feedback. Therefore, I cannot conclude and rule out that the deep layers might be keeping the information of the prediction error.
Okay, thank you very much.
So the opposition will now be continued by Professor Roebroek, Professor of Brain Connectivity and Computation at our department at the Maastricht University.
Thank you, Prorector. Dear candidate, I also want to briefly add my congratulations to ones that my colleagues already gave for a very, very beautiful thesis, very comprehensive work. And particularly, it's been said before, but it needs to be emphasized, the way you went over multiple scales in neuroscience and also across function and structure is very impressive, and I want to extend those congratulations to your promotion team. In the interest of time, I'm going to try and keep my two questions short, because over many scales and over both function and structure, you need many methods, and you have used quite a few methods. And I have two actually, methodological questions. The first is about chapter 2, where you did some beautiful functional and also effective connectivity analysis. And I want to focus there on the correlation analyses. Could you very briefly explain your choice there of choosing lagged correlations from one area to another, and also to try and derive the directions of influences and how, for instance, differences in hemodynamic effects in different regions might interfere with that, what your thoughts and the reasons for your choices were there?
So, highly esteemed opponent, thank you for the compliments, first of all, and for the question. Yes, so there are reasons why we did choose at the end to go with the lag-based functional connectivity analysis instead of other methods. So, first of all, I acknowledge that there is a limitation in terms of different, the lag due to the hemodynamic response. So that's impossible to, of course, hide. It's clear is there. However, this method has been used already in the past and even embedded into computational model, as it's shown that even if we are sluggish with a hemodynamic response, it can still capture the difference in temporal dynamic between two brain areas. So by saying that, the analysis that supports this statement is done by using an autocorrelation function, and that I compute myself, and it was also computed in the past, and it's shown somewhere in my supplementary material, that allows us to choose at which lag we are still seeing a meaningful information while we do change it. Then, so this was one way to assess whether this could give us something out of it. And then second, of course, it's embedded into these computational models. And the one that I used here is a physically inspired one called the arrow of time, or generative effective connectivity of the arrow of time, if you want to have the long spelling. And there they show that embedding such an information, more than having only the functional connectivity, can actually fit better with the empirical data. And I did even try myself to test, because this method was developed to use it as a measure for the resting state and different disease or pathology of the brain. But now we did apply on a task fMRI and on a perceptual fMRI. And we still see that having such a lag-based information, or this covariance matrix as you want to call it, had a role, and a prominent role. The data were fitting better, and we could reproduce the phenomena that we were seeing. Third, we could have chosen the closest, of course, methods is Granger causality, and you have been working on it a lot, of course, and I did use it and compared the result. So first, from an analytical approach, mathematical basis, just to, of course, be sure that we were at least capturing part of the variance that Granger causality was capturing. However, the result did not converge 100% because there were part of the connectivity that were different. On the largest end, they converge. Small details come from the fact that, of course, Granger causality is more comprehensive in a way, and it takes into account the information of what is happening instantaneously as a variance, and take this apart, right? But we didn't choose it to use it because we want also to be coherent with our paper. We did use this computational model that has already this property embedded, and we want to let these methods talk to each other. So this was another reason. And finally, we even thought to use dynamic causal modeling for such a case. I'm not hiding that maybe I still I will do it in the future. I didn't test it, but given our first result that highlight a few brain area in the network, I think there is a value of trying to do even with DCM.
Right. Thank you very much. It's a very comprehensive answer. I'm looking for one more very short answer on your structural imaging, if I may.
If both the answer and the question are really short.
I will keep the question short. I give you the challenge of giving the short answer.
Yes.
You did beautiful structural imaging in chapter 5. You had postmortem data at quite high resolution. However, cortical layers are sometimes less than 100 micron. What is the ultimate resolution that we need at least ex vivo, to see all the layers?
Yes, so I think that being already below 0.3 gave us some information about the big structural changes.
But what is the ultimate resolution needed to see everything?
Okay, then I need to use a little bit of a rule of thumb. If you need to have 6 layers and we want to decode all of them, assuming that they can be decoded in terms of the contrast that we have, I will divide my average thickness, that is 2 millimeters by 6, so 0. And let's make it double, so 0.1.
If they were all the equal thickness, but we can discuss that over the drinks.
This is a very short answer that I could come up with.
Thank you very much.
This is the reasoning behind, I think.
Thank you. I'll give it back to the prorector.
Great, thanks. So the opposition will now be continued by Dr. Kirillina, expert in neurophysics at the Max Planck Institute for Human Cognitive and Brain Sciences at Leipzig.
Yeah, thank you very much. Dear candidate, I would like first to congratulate you to the impressive thesis and also thank you for the excellent presentation. I can only echo my colleagues and say it was remarkable to see the PhD project that bridges together such a wide range of advanced neuroimaging and analytical techniques and all executed with such a high quality. One major strength of your work is also the ability to link between their functional structure and between the post-mortem and in vivo MRI. I found it very impressive. And I think it's a crucial step towards advancing our understanding of brain organization. And you have shown that it's not trivial, and you developed multiple tools to establish this link correctly. So congratulations to this significant achievement. My questions relate to the final chapter of the thesis because it's closely aligned with my own interests. And I want to challenge your proposition number 3, where you're saying that bridging postmortem microscopy and in vivo imaging enables a more complete structure-to-function coupling in the study of the human brain. And I fully agree with that, but I think so what I want to challenge is how exactly we are doing this link. So you aim to link the function structure in your final study, and you analyze the cortical profiles of relaxation rates recorded in vivo and postmortem and compare them to the amplitude of functional MRI signals. Here my question is, you used the mean signal of epi images and compared them with the quantitative parameters, and not their amplitude of fluctuation. That's something what we typically use in MRI in the resting state. So could you explain your rationale of analyzing the mean signals, which probably also reflect the structure, and not looking at the amplitudes? And in case you look at amplitude, perhaps you can speculate, do you think this would add information, looking at the profile of amplitude of the fluctuation, maybe closer reflecting the function?
A highly esteemed opponent, thank you very much for the compliments again and for the question. It is an interesting point. So the rationale, so you were asking why I did use in chapter 5 and the microscopy the mean of the resting state fMRI, and whether the amplitude of the fluctuation of the resting state, right, would be more beneficial instead and gave more information. So yes, I totally agree with your point. And first, we did use the mean of the resting state because we want to have a methods or a summary of the resting state that's very simple, the simplest as possible. And this basically, just doing the average, I saw other people in the past doing as well, like Peng et al. And it was very crucial for them when they did study different laminar profile from people with a specific disease that now I don't remember. So, and they could already capture some of the variants there. So basing on this result, and I did try to use this as a first step to bridge and see how this information can be useful. However, I do agree with you that looking, like we need to progress. This was just the first step. And we did assess that the R2 star value are showing the same property across the 3 data set. The postmortem that only capture the microscopy side of the tissue, the in vivo MRI that has the microstructure, but also, of course, the vasculature that gave rise to our contrast a lot, and the resting state that is technically the basis of our functional data. Therefore, in the future, I think we need to use some other measurement derived from the resting state. And the amplitude of fluctuation can be even more beneficial if we then try to link it with the neuronal types. And this is a bit of a stretch, of course, but in this the thinking, I will still see that, okay, if there are different neurons, that they have different frequency and so on, there is a big sea between the two things, but maybe one day we can bridge it, and then using methods that can capture such information would be more beneficial. Yeah.
Thank you very much for this answer, and I'm very happy that you are planning to do the next steps, perhaps to following up on that. So you demonstrated these different slopes which you record in postmortem and in vivo, and pointed out that probably, so I think that's a particular strength of the work, that you really leverage the power of quantitative MRI and really compare the amplitude of the response, of not just the shape of the profile. And you argue that this vascular contribution, which goes kind of oppositely to the myelin would increase to the deep gray, from pale to white matter surface. And you are saying the vasculature gives the contribution, which has an opposite shape. Do you think you could quantitatively estimate these effects based on the histology we have? Or perhaps you experience some limitation of the current histological approaches, and you would wish something from the current anatomical atlases of histology, which would be needed for that step.
Yes, if I understood you correctly, you are asking if we can compare the slope of what we found in vivo and ex vivo in postmortem data about the profile of R2 star. And this is actually a computation that we did in the thesis, is in the chapter. Of course, it comes with big limitation, like for instance, the R2 star contrast does change in a postmortem sample versus having an in vivo sample. So this is the biggest limitation that we have to accept in our data. And but hopefully, like compared to a T1 contrast, the T2 star was found or demonstrated by other colleagues that is less affected by fixation. And also, I think this is supported by the fact that the range of R2 star that we define here are very similar. And then we did the assumption that maybe this 2.8 factor of difference between the two slopes is still meaningful for us.
Thank you very much.
Thank you. So now the opposition will be continued by Professor De Weert, Professor of Neuroscience of Perceptual Learning and Attention at our department at Maastricht University.
Dear candidate, I also want to extend my congratulations for your work to you and your team. In the interest of time, I'm going to go straight to my question. One of the things that I read in your very interesting thesis, I found very surprising. Naively, I would have expected in the ambiguous perceptual state, right, that in the superficial layers activity would go up, right? That you would, and you, in fact, what you see is that you don't see this. And you have an explanation for this in your thesis, which I try to understand, and I don't completely do. My intuition is that it has something to do with the kind of feedback that is going down from MT, maybe other areas as well, to V1. Could you explain to me a little bit more Yes, we are talking about your explanation on page 75.
Yes.
Could you explain to me a little bit more what your view is on why you didn't get this, maybe anticipated effect of increase of activity towards the superficial layers?
So, highly esteemed opponent, thanks a lot for the compliments and for the question. So this is a crucial point of the thesis indeed, when we did look into the laminar profile. Maybe I wouldn't have bet directly on the role of superficial layers, but having like a equally likely activation both in deep layer and in superficial layers, because we actually don't know which one of the two can be receiving the feedback, if the feedback is coming from, of course.
But you didn't find either, right?
No, yeah. I didn't. And this, I think, goes together with the acquisition technique that we did use. So, as I highlighted before, there are limitations due to this draining vein. And usually, this increase that comes with the bold contrast that we are using. So, a different choice might have given us different result. So the fact that the laminar profile become basically flat, I did interpret in this way as the following. So when you have a feed-forward condition, and most of the layer fMRI study actually target this specific condition, you receive the biggest input in the middle layer from the thalamus. And this drains up to the superficial layers directly. And it gives you this increase that is the most, I think, replicable result in bold layer fMRI. However, in the feedback condition, if there is not such a strong input, right, because the thalamus is not sending, the motion is not on the screen, right? So those neurons that are in between the inducer are not receiving input. So we start already with a situation where our signal goes very low, is very low. By saying that, maybe one explanation could be that the signal intensity is so low that doesn't induce draining. So if the activity, so there is, and this is of course overly interpreting the data because I cannot say which layer is active here. They can be all of them because I do see that they're not zero. So there is something going on. And if we think for a second, if the input would have gone to the deep layer, maybe there is no reason, and if it's low, there is no way that it will reach the superficial layers and give us this increase towards surface. This is in the hypothesis that the feedback reached the deep layer. I think I can, of course, not rule out directly that the superficial layer is not receiving the input. This is a limitation. And in that case, I don't know how to explain straightforwardly. I do need and might want to have a high resolution vasculature imaging behind because I want to understand where the blood is going. If the signal reaches the superficial layers, then somehow if I know the geometry, I could, we can better correct those data. But at the moment, that kind of imaging was not available for us, so we did not further explore this possibility. If that makes sense for you?
Yes, it does. It actually has implications for many studies also that use other visual illusions where you actually see very small effects.
Yeah.
So yeah, it's an interesting perspective. I think in the interest of time and saving time for my colleague, I think I will just give the word back to the pro-rector and there will be more time for the next discussion. Thank you.
Thank you very much. So now the opposition will be continued by Dr. Zenden, expert in computational neuroscience at Cognitive Neuroscience Department at our university.
Yeah, dear candidate, I would also like to extend my congratulations to you and your supervisory team and your family. I very much enjoyed reading your thesis. And what I particularly enjoyed, apart from the really good science in chapters, in the empirical chapters, I also very much enjoyed reading this reflective approach and this meta-science perspective that you had in the general introduction and general discussion. And I would actually like to focus a little bit on this now. So we had three interesting components there. The one is the argument for deep sampling, then for the reduction or isolation, and then the multiscale. And since I only have time for one question, I would like to focus on the reduction. So I find it a very, very smart way to tackle complexity. But I was wondering what you think about to what extent this approach forces you to make the assumption that the brain or the mind or both are modular, to what extent you would agree with these assumptions, and then also to reflect on to what extent that affects what you can derive, conclude, understand if you take such an approach, depending on how much you believe these assumptions.
Yeah, so highly esteemed opponent, thank you for the compliments and for the question. Yes, so we are here discussing about the reductionist approach that I did use in the thesis, and you're asking me whether I think that the brain is modular. So if we define that, like, how do we define modular, right? First of all, so we need to clarify that. And if my understanding or what I can define right now, modular might be due to the function of so the brain area are already like a modular way, and there are different levels of modularity. And it can even go down, of course, to the cortical layers and the cortical columns. So I do believe that the brain is modular in a way, so that there are groups of or clusters of neurons that are doing a specific computation. And I do think that we can grasp them with the fMRI due to the size of them and if they act together. In some extent, of course, not like fully. And I think that we always need a kind of pairing with the other methodology that, for instance, insight from electrophysiology might help us to understand how modular are these features that we are trying to decode with the fMRI. Yes, so I do think that the brain is in some extent modular and that we can use such a feature to understand how it works.
Okay, then I like your answer. I will push you once more further, but taking your analogy of MC Escher and the painting. So by studying this one painting, how much can we learn about MC Escher as an artist? And how much can we learn about the painting itself, never looking at any of the other paintings?
Yes, so this was actually really the point of why I decided to have this picture, this beautiful frame here in the thesis. I think that looking at one, of course, painting, it's this reductionism approach, right? So we cannot fully learn what the author wants to communicate through his art pieces. So, but in here, we can understand that something is challenging the idea of gravitational forces, for sure. And you see by different stairs and stuff. So this effect is a statement that we can have and what we can learn. But of course, it's reduced to this painting, and we need to have like explore multiple if you want to bring all the pieces together and understand the artist. And in a similar way, like that's what we did in the brain. We know that we are limited, that we are only looking to one phenomena, it's vision, visual perception, bistable perception, so very specific things. But going through multiple scales also gave us this interesting perspective and let us, I hope, understand a bit more how this works thoroughly. Yeah.
Okay, thank you very much. Then, of course, the deep sampling. I found you make a very nice case about the deep sampling. There's, of course, also the large N approach. And I was wondering to what extent you think these are separate or complementary, and whether you can see in your work a concrete way of leveraging large N for, together with your deep sampling.
Yes, so I think that very briefly, we can maybe complement them, and in the future, using...
You may complete your sentence.
Yeah, so I think that in the future we should actually go for a larger N, but still having this deep sampling approach, so going across scales. So one of the other doesn't exclude the possibility we need to combine them. Yeah.
Thank you very much.
Thank you. So, Alessandra Pizzuti, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if there's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Alessandra Pizzuti, the degree committee here present has assessed the quality of your thesis and your defense, and in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Goebel is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Oh, this is the Dutch version, sorry. I could do that too, but I think we switch to English. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Alessandra Pizzuti, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. So, this was a performance. My gosh. Congratulations, Alessandra. I'm so proud of you. This was one of the best theses I have seen, really. Both the thesis and the defense. And I want to congratulate also your family. No, I don't manage in Italian. But I can tell you, you can be super proud of Alessandra. This was really a great defense. And actually, I want to extend my congratulations to the or my thank you to the committee members. That were questions at all levels, highly multi-scale questions, but you managed, right? And you integrated them even, which Professor Di Martino wanted to have also, right? So I think it was really a super performance. And I want to also convey you the congratulations of Judith. I integrated in my laudatio, but I read her poetic sentences which I really like. Listen, from Judith. Layer by layer, I got to know you and how your mind operates at ultra-high field strengths. Layers of great programming and analysis skills, meticulous project organization, and scientific talent intertwined with a fun personality across multiple scales. I enjoyed every moment working with you. This counts for us both. Regardless of whether we were figuring out hidden sequence parameters at the scanner or hidden brain patterns in your data. Congrats, Alessandra, you did an amazing job.
Thanks a lot, Judith. Thanks so much.
So what we usually do in such a laudatio, we look a bit back and look how things have started, right, and emerged. And actually, it started with obtaining a PhD position in the context of a European Training Network grant, the USNN, the European School for Network Neuroscience. And I looked for a PhD position, put an ad out internationally to really want to be open-minded, to find someone who fulfills these roles. And I was actually very careful to select because the topic I knew will be extremely challenging, and it goes to the deepest of my heart from the topics. So therefore I said, let's have some time and find someone, right? So when then you applied, it was actually, not a neuroscientist, not a psychologist, it was a biomedical engineer who caught my attention. your CV and also with your experience, programming skills like you did mention, but also you had already experience in ERC as a research fellow, and yeah, you wrote also, described yourself well, had this passion, wanted to really change something in the world. So I really was fascinated by what you wrote and your background and skills. And this, in the interview, was the first time where I then could also experience you, your spirit, your dedication, your motivation to do something great. And I thought, I stopped interviewing. People said, you should go on. No, I'm fine. I didn't tell you that. But I stopped interviewing because I was decided, basically. And how right was I to give you this position? This, what happened here today makes me so proud. It was a journey with a bit of ups and downs, but mainly ups, I would say, right? So, some things look at the moment a bit tough, like getting a paper rejected or so, but at the end of the day, you published everything. The last chapter is submitted, so in principle, big success. And you had time when you were bored, as you told me, to do lots of side projects, right? So one is now just another era of time in Parkinson's disease with one of your close USNN colleagues and friend, Fatime, and this is just published, right? So it's amazing what you did as side projects. Also, when you went to the HBM at the hackathon, then people told me, she can do all this AI stuff, right? And lots of, yeah, I do this on the side, I'm just interested in that. So what you did on the side is already a thesis, right? Actually, yes. So therefore, you have today 2 theses. When you started, it was, of course, a tough time. It was still COVID, right? And we couldn't meet in person at the beginning. And yeah, and since the grant was awarded to Brain Innovation, it was great to see how Claudia supported you with all the managerial aspects, moving, and so on. And I want to thank her for all her support on this. And also Brain Innovation, everyone there, yesterday I met them, everyone is super proud of you, right? And they really are happy to be here. I saw many of them here today. So this is really wonderful. And yeah, what came to my mind when I looked for adjectives, I will not say them all, there are many, 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 but one word, outperformer. This are you. You are really an outperformer, in the most positive sense, I should say. Yeah. And then, yeah, you were super productive, but I want to also say that at the beginning, how fast you learned was for me breathtaking. And you had really good people surrounding you, which like to support you. And I want to thank here especially also Renzo. He was in the first years very important. And the most important person, probably next hopefully to supervisors, was definitely Farouk. Farouk became a mentor for you, right? Yeah. So this is incredible what he did. And I can tell you that he was also your spokesperson. He came sometimes to my office, Rainer, I think maybe, right? And I loved it. I loved it. Wow, this is dedication. This is mentorship, right? So this was really great. And yeah, what I also liked a lot, which also many peers told me, that they admire your brilliance of discovering details in data. And then to not give up and dig deep. One case was a setback, namely you discovered a problem with the scanner, with the 7T, and no one listened first. And you said, Rainer, here is something different. And I said, yes, you're probably right. We have to convince the others, right? At some point, it turned out there was a hardware failure, right? And basically, you were one of the first who realized there's something really wrong. And this shows another big talent you have. So now I have to skip, otherwise I'm standing here a bit too long. I just want to say also how proud I am of your publications. When I see the cerebral cortex cover, I'm always smile, right? This is wonderful, really wonderful. Yeah. And I also want to thank you for another side project you were involved, and still are, the Meta project, Happiness project. And I think we had some very nice discussions suddenly also about what happiness is in our more mentoring kind of meetings where we talked about career and other things. And that was really nice how dedicated you were, and you made basically the poster and presented this at OHBM. And so that also showed how many topics you did next to this very difficult and challenging work. Really impressive. You also had your USNN family and also did their meetings, first online, later on in person. I remember the very nice meeting we organized in Maastricht, where all your fellow PhDs came to Maastricht. We had a nice dinner, and it's always fun with you at dinner. The last one where we were with the company out, just last year, was your birthday, right? It was the 30th birthday?
Yeah, 30th.
A very special day. and I'll never forget this, yeah. Question is, how does Alessandra relax, right? What do you do to compensate? Side projects, yes, but there should be more. And of course, I know that you love art. So for example, we both share this passion for Escher. And Lars mentioned this Gödel, Escher, Bach, which was for me a breakthrough work when I was maybe in your age, roughly speaking. But I heard you also do sports, and you like padel, is that true?
Yes.
Yeah, I see you have good spice. And so that you became kind of more passionate to do this. Mm-hmm. It's a trend sport, I read. I did not even know it, right? To be honest here. Yeah. So I think you will have a great future ahead. And like in your thesis, sometimes things you plan, and we plan together somehow, or advise you at least, some things work out, some not. And sometimes it's even out of your control. For example, when there are changes in the US, then plan A simply doesn't work, doesn't matter, right? You are so smart, so brilliant. You find other plan Bs which might be better than the original plan A. So I look very much forward to you, and I have to say that the feelings I have towards you can be best summarized by the German word Doktorvater, which is a word which we use in Germany for PhD students. In your case, I mean it quite seriously, and I want that you never forget that you have an academic father now who is always there for you and who wants that you flourish in life. Thank you.
Thank you.
Thank you very much for the heartwarming laudatio. So, Dr. Pizzuti, also on behalf of our department and the Faculty of Psychology and Neuroscience and Maastricht University, I would like to congratulate you with the degree you have acquired. And you really did an amazing job, really impressive to see both your thesis and how you defended your thesis here. And I, of course, would also very much like to extend the congratulations to both supervisors and also the extended supervisory team. And tantissimi auguri anche alla famiglia. Potete essere molto fieri della vostra dottoressa Pizzuti. I would also like to thank the audience, and both here and online. And of course, I would very much like to thank the committee for their inspiring and interesting questions and the discussion that followed. And I would like to also especially thank the external committee members for participating in this discussion and bringing their expertise also here. And yes, I think then I thanked everybody I wanted to thank. So just in general, thank everybody. And now the audience will be asked to actually go to the reception, which will be outside in the courtyard. And we will still, as a committee, stay here for pictures together with the online committee members. And then afterwards, the idea is actually to take pictures also together with the family on the stairs, like in the main hall. And then after that, we all go to the courtyard where everybody can give the congratulations to our new doctor. And hereby, I close this academic session.
Yes.
Somehow.
Don't forget.
Yeah, forward, so half. Kom je niet. Please come a little bit forward. So.